Our next speaker is Shen, professor of informatics in biostatistics and epidemiology at University of Pennsylvania. He will be presenting the work entitled Enhancing Alzheimer's Research with AI and Informatics, Strategies for Mining Brain Imaging Genomic Data.
Thanks for, thanks for the introduction, and also thanks for having me here. I'm going to talk a little bit about the AI and the bioinformatics strategies for mining brain imaging genomics data, hopefully to advance Alzheimer's research. So that's the disclosure. And so we know Alzheimer's disease is in the US, it's a national priority, right? Research priority. The major challenge is there's no drug that can modify the disease. Although there's some recent development, right, in terms of the drug, there's a few drug approved recently, but it's still a major challenge. But the thing here is we know the major hallmarkers, right? So you can have the amyloid plaques and neurofibril tangles, so-called ATN classification system, right? Amyloid, tau, neurodegeneration. All these actually can be captured by imaging. So that's why we want to use imaging as phenotype, right, to study genetics. Hopefully, the different modality can be corresponding to different subtypes. In that case, we can identify subtype specific, right? The genes and the proteins, and as targets, hopefully we can find a drug to modify those later on, right? So Paul Thompson and I wrote this review paper on brain imaging genomics. So basically, we have all kinds of these biobank data, right? Starting from the genetics, whole genome sequencing, genotyping, and also molecular data, right? Multi-omics data, and at system level, there's imaging data and fluid biomarker, and then there's a phenotypic outcome, say cognition behavior, right? So basically, brain imaging genomics, we try to figure out the relationship between these modalities, right? So we kind of summarized a few problems. The first one is since we are using imaging as phenotype to study genetics, you can extract all kinds of imaging features, right, from MRI scan, PET scan, and then how to prioritize those, right? So basically, we want to start with the imaging phenotype with a stronger genetic basis, right? Meaning the heritability should be high, right? So that's why there's a heritability estimation issue. And another thing is that we want to figure out the association between imaging and the genetics, right? So that's a kind of a majority study, right? And then there's another kind of problem is you want to integrate the genetics and the imaging, hopefully can better predict the outcome, right? And also, sometimes we want to figure out maybe the biological pathway from genetics to molecular signatures and to imaging phenotype, and then to the phenotypic outcome, right? So try to figure out the biological pathway. In that case, if you see evidence at different levels, then that's reassuring, right? So I'm going to briefly talk about three recent studies. Actually, all has been completed by my excellent PhD student, Jing Shen. He's actually in the audience. So the first one is, Jing Shen actually developed this giant, it's a brain atlas. The idea here is, we know there's a lot of atlas, right? The AAL atlas, Glasser atlas, right? So Schaffer atlas. Many of those are defined based on the anatomical knowledge, or maybe the functional data, right? They are not optimized for genetic study. So here, what we want to do is, we want to define the atlas. Hopefully, using that atlas, we can extract more heritable imaging phenotypes. So the idea is very simple, actually. We use UK biobank data, right? And then we kind of did a heritability analysis at the voxel level, right? We're using this LDAK to do the heritability analysis. Use this Reven's map, the gray matter voxels, white matter voxels, right? And once we have the heritability map, you can do segmentation. So here, Jing Shen actually developed this new kind of 3D Gaussian mixture model here. So the idea is we want to group voxels with similar heritability together. At the same time, they need to close to each other. So That's why we have this easy model here, right? So if you look at the results, basically our segmentation, right? Based on our segmentation, you can look at within the region defined by our atlas, the voxel level heritability are more homogeneous, right? So these are actually tested on an independent data set, not the data actually we used to generate the atlas, right? And another thing is you can extract the regional level heritability. So we compared our segmentation results with just the watershed and with the existing atlas, the Muse atlas. We can see our phenotypes, regional level imaging phenotypes are more heritable. Then you can do the GWAS analysis or polygenic risk score. We just heard about that. Then, so basically using the GENT atlas, we can find more hits, and then the more variance can be explained, right? So basically, so these are some results. So these are voxel levels heritability. And you can see the heritability measurement and also the p-values, right? Based on that, you do the segmentation. We extract 50 GENT regions, and then we can generate some genetic landscape of these GENT traits, right? So you can see these are the chromosomes, and the color indicates where are these GENT regions come from. So these are the color, right? And then we also look at the kind of like the shared genetic basis between these GENT traits with some common traits, right? Like the weight, height, BMI, so you can see the relationship. And then these are just some functional annotation, and for some of these GENT regions and the corresponding genetic kind of variants that belong to different, say, 3' UTR, 5' UTR, maybe exonic intergenic regions. So these are just some landscape. And then we can calculate the genetic correlation between these GENT traits and also phenotypic correlation. So you can see they are similar, but not exactly the same. So hopefully in the future we can use that to define the traits, identify interesting, say, AD genetic kind of biomarkers, right? So that's the first one. And the second one is we want to focus on trying to find the imaging genomic association, right? So we know this is an early like ADNI study. We use image structural MRI like as phenotype, then you can find a lot of hits, right? So many of those are missed by the case control study, showing the promise, the statistical power of imaging phenotypes, right? So you can actually map these onto the brain to see all the genetic effect of this SNP, and you can map the imaging phenotype into the genome, so you can see the genetic association for those phenotypes, right? So we also did this. These are ADNI studies. Since ADNI is a part of Enigma, right? So some of those are Enigma. So using those quantitative traits, not just the imaging, but also say CSF biomarker, like memory score, we actually identify a lot of interesting genes, right? So that shows the power of, has some scientific clinical impacts. So like from the Enigma and the UK Biobank, they actually share a lot of summary data, and many people are using those summary data for subsequent analysis, right? So that's very useful and valuable. And in this study, we actually use the amyloid quantitative traits as phenotype. We identify a gene, and this actually has been used as a kind of knockout, like build an AD mouse model. So that's pretty impactful. So we also identify some candidate genes. So, and then here a little bit about machine learning. We know that CCA, canonical correlation analysis, has been widely used in imaging genetics to try to figure out these bi-multivariate correlationship, right? So you have all these genetic markers. You can calculate a genetic component, which weighted sum of these, right? We can also calculate imaging component, and the CCA try to maximize their correlation. You can add some constraints to reduce the risk for overfitting, like the lasso sparse constraint. In this work, we actually, we actually added this additional preference matrix. Basically, if I have some prior knowledge, I know, oh, this imaging biomarker is related to that SNP, so I encourage the model to find that one, right? So you can define a preference matrix. So this can be modeled here. So here just a proof concept study. So we looked at some phenotypes. These are somehow related to AD, some are not related to AD, right? So these are the, say, cognitive scores, imaging AD biomarkers, free surface measures, right? So these are summary statistics. In the genetic side, we looked at some AD SNPs and some SNPs, random SNPs, right? So they are not relevant. Then we applied our model. So if you look at our results, the traditional CCA and the sparse CCA, the training is okay, but if you apply it to an independent data set, you don't see the correlation at all. But using our preference matrix guided learning, actually we can see higher correlation, right? If you look at the biomarkers, so these are the CCA, SCCA, one is the imaging, one is the genetics, and you can see everything is involved, so that's not helpful, right? But using our methods, we only identify, say, AD relevant biomarkers, right? So that's another thing. And so finally, I want to look at the last one is that we kind of like want to integrate all these genetics, multi-omics, and imaging outcome together. So ideally, we want to find something like, say, the SNP is EQTL that can regulate the gene expression level of this gene, right? We identify a gene, and this gene is highly expressed in certain AD-related cell types and also AD-related like a tissue, the region. Like here, this is entorhinal cortex. We know it is actually related to the dementia outcome. In that case, you can link all these things together, right? So how to achieve this goal? So first, we actually use UK biobank data here. So you can do imaging genetic analysis. We have a lot of imaging genetic findings, but these findings may not be related to AD, right? So we actually look at some AD summary data. You can do some co-localization analysis. In this case, you can kind of prioritize those findings, extract a subset of those that are co-localized, right? The genetic finding in terms of these two phenotypes. One is diagnosis, the other is imaging, right? So now we have that. And then we actually did some mediation analysis, try to figure out, maybe some imaging phenotype can serve as mediator, right? That can mediate the genetic effect on AD. In that case, actually, we can narrow down the search scope, right? And once we actually identify the entorhinal cortex here. Then we have the genetics imaging outcome, and then since we are missing omics, but there's a GTEx data, right? There's EQTL data. We can actually do the co-localization analysis to link these things together. In that case, we can identify this BTBD16 gene, right? And then we did some like additional analysis, try to see, this gene is actually highly expressed in the cortex and also in this specific cell type, right? So in that case, we can link everything together. So it turns out all these things are related to amyloid and AD, right? So basically, that's it. And so here's a review actually we did in 2020, and Jingxuan actually wrote another one. If you are interested, it's up to date, just published, and try to figure out the relationship between the genetics and outcome, multi-omics with outcome, and multimodal imaging with outcome, or maybe some causal relationship involve multiple kind of scales, the data, right? So yeah, this is what we do actually in my lab. And our goal is try to identify new AD biomarkers, AD subtypes, a better model disease, hopefully to identify candidate targets and drugs, right? So finally, I want to thank my lab members and also NIH and the NSF for supporting our research. Thank you.